Our project is over the synthesis of silver nanoparticles with sugars using the Tollens method and sodium borohydride. I'm Ashley.
And I'm Lynn.
So we'll be comparing a chemical and green process of synthesizing silver nanoparticles in order to find a safer and environmentally friendly procedure that doesn't require the use of harmful chemicals.
About silver nanoparticles, they are increasingly used in various fields, including medical, food, healthcare, consumer, and industrial purposes due to their unique physical and chemical properties. They are widely known for antimicrobial properties against bacteria, fungi, and viruses like influenza A and HIV, and currently COVID-19 and SARS-CoV. And for synthesizing nanoparticles, there are three ways. And in our project, we are focused on the chemical synthesis and the green synthesis. The green the chemical synthesis appear to have low cost and high use. However, the use of chemical reducing agents are harmful to living organisms. In contrast, the green synthesis is shown to be simple, cost-effective, and especially environmentally friendly approaches. And there's a picture of our successfully experiment that we conduct with chemical synthesis when the sample has a color of a yellow, and then below that picture is when the sample gets aggregated, it turns to a dark color. And next to that is three vials with the green synthesis that are conducted with different sugars to see how it will affect the size of the silver nanoparticles. About the antibacterial, we are going to conduct experiments against the bacteria called E. coli and using the Kirby barrier test. The silver nanoparticles will attach to the cell wall of the bacteria and restrict many processes that are needed for the bacteria to function and reproduce. Our goal is to get the size of the and nanoparticles as small as possible for the better effective against bacteria.
And then for our analysis, characterization, and kinetic studies, we'll be using ultraviolet visible spectroscopy. So UV-Vis will help us determine the range of sizes that our nanoparticles will be in, and it can also help us monitor the rate of aggregation.
For the chemical synthesis, we're using sodium borohydride for our chemistry reducing agents, where we will conduct experiments with different concentration of sodium borohydride to see how it will affect the size of the nanoparticles. And the sodium citrate is also used to stabilize the nanoparticles from aggregation. And for the green synthesis, we are synthesizing nanoparticles with three different sugars: glucose, sucrose, and starch.
And for our UV-Vis data, we've calculated the average absorbance and wavelengths, and we compared our chemical and green results by putting them into graphs that show the peak absorbances. So for this project, we want to find ways to synthesize silver nanoparticles safely and effectively, and we will also begin our antimicrobial study with E. coli to see how effective our nanoparticles are against bacteria.
And we want to say thank you to Dr. Frost, Dr. Scott, and Dr. Wicker, and our TA Ryan for helping us and giving us the opportunity to do the experiment.